Welcome back to the lecture series in application of nanomaterials in agriculture. So, in the previous class, we were discussing that how cerium oxide affects the muscle physiology, cerium oxide nanoparticle. So, in that context, I told you that there are intricate physiology of the muscle. Once you understand that, it will be very easy for you to appreciate that how this whole contraction profile, force generation profile is being modulated by the presence of an antioxidant nanomaterial. And apart from it, I told you that we will be talking about some of the tools, microfabricated, nanofabricated tools, which are being used to understand muscle physiology and as a diagnostic tools for muscle growth and muscle development and their respective force generation. So, let us catch up where we left in the last class. So, in the last class, if you remember, so this is where we were. So, we talked about the development of muscle, and there we talked about the alignment of the actin and the myosin filament, as you could see. So, now today we will talk about these two aspects, sliding filament theory and excitation contraction coupling apparatus, and how cerium oxide is influencing it. So, when you look at a myotube, cross section of a myotube is something like this, which becomes myofiber. So, I told you that there is a very interesting arrangement of the actin and the myosin filaments. So, something like this, and if you go to any textbook referring to actin myosin filament arrangement in skeletal muscle, you will come across this kind of wonderfully drawn pictures like this. I am not introducing the colors yet. Now, soon I am going to do this. Okay. So, now you see there is a pattern here. So, if I had to put the pattern, so it looks something like this. So, I am translating these lines, which I am making red now. Okay. So, these filaments, what you see, which I am now coloring them in red, these are the myosin filament, and the myosin filament, as I told you, changes their protein subtypes as we mature. So, some of them are thick, some of them are thin, and depending on that they have a different force generation potential. And the one now I am putting them in green, blue. These are the actin filaments, okay, which are much more thinner. Okay. So, now this is how this arrangement of myosin and actin is being orchestrated by nature. And the lines what I am drawing here, the jagged lines, these are the Z lines. And in between these myotubes, you will see some kind of gaps like this. And then again another myotube starts, and this will continue. Okay. And we will talk later about those kind of jagged portions or the grooves which are formed. And here you are having the actin filaments, here you are having the myosin filament. Okay. If you look at this picture pretty closely, you will observe something very neat. This picture is more like this. Here is a myosin. The myosin has these kind of heads top of them. These are called the myosin heads, something like this. Okay, and on top of that you are having these actin filaments rolling like this. Now, if you look at the actin filaments, these are supramolecular assemblies of multiple proteins, something like this. And in a normal condition, this actin filaments are in proximity with the myosin heads, but they are not in touch. They cannot interact with the actin filaments because of the blockages which are present out here. There is something called, these are different proteins present on actin filament: troponin, tropomyosin complex. There are multiple proteins which are involved in it. That is not important. Now, this troponin tropomyosin complex prevents the interaction of actin and myosin, especially the myosin head and the actin filament. Actin filament and myosin head. Okay. Now, when there is an increase, all of a sudden there is an increase of one of the cations, which is calcium, Ca2+. This blockage between the myosin head and the actin, because of the troponin and tropomyosin complex, is this complex gets disintegrated transiently. As soon as this complex, troponin tropomyosin complex, gets disintegrated in the presence of calcium ions, the myosin head binds to the actin filament here, and it does something called a power stroke. So this head moves like this, and when it moves like this, there is a contraction which is observed in the actin and the myosin filament, and this is where the force generation is. So the force with which this myosin head is moving is what we call the muscle force. All of them move like this, and this is what generates the muscle. Now, for this movement, which is technically called as power stroke, you need ATP. Okay. Now, does this strike the bell in your mind? This is, now let's correlate what is happening when you talk about the presence of ATP there, and we are talking about the ATP content here in the presence of cerium oxide redox couple. So you realize, in order for a process like this to occur, you need to really appreciate how one simple autocatalytic redox couple can change the muscle performance. So this is where this whole ATP thing comes. So if you have more ATP, then that power stroke is going to will be able to enact its action better. Okay. And of course, I will request you to go through the sliding filament theory, which is just now essentially I taught you. And there is another one which is called excitation contraction coupling apparatus, which is present somewhere out here in this zone, which essentially regulates this calcium, because this calcium cannot remain there forever. The calcium has to be dumped there, and it has to be pulled back in a fraction of a moment, and that is done by an organelle called sarcoplasmic reticulum. And one more thing what I wanted to highlight here, as you must have seen that these muscles have higher power generation. It means the mitochondria which is involved, which is lined up this part all over, those mitochondria are far more efficient in cerium oxide treated animals. So, this is the part what I wanted to highlight, that a simple molecule can make a whole range of difference in the performance of muscle. With this brief idea about muscle biology and the role of cerium oxide nanoparticle, I will move on to a diagnostic aspects approach by which you do not have to kill any animal or sacrifice any animal, and yet you can understand the muscle physiology better. What our nanotechnology has to offer in terms of diagnostic tools. So, there we will be talking about, as we promised, about nano and micro cantilevers. So, okay. So, since now we have talked about the force generation out here, power stroke and the force generation. So, we needed a tool to study force generation of the muscle. Of course, you can do it in vivo in animals as well as you can do it in vitro. In vivo, much of the tools are known to the exercise physiologist. In vitro, there are upcoming tools which are using different kind of nano devices, micro devices to study these kind of force generation. So, here I will talk about one such micro nano device which follows one of the techniques what we discussed very early in the course, how we can develop nanomaterials. We can develop nanomaterial from bottom up approach, from top down approach. Okay. So, today we will talk about one of the micro nano device which is developed by top down approach. Okay. So, today we will talk about fabrication of micro and nano cantilevers for studying or for diagnostics of muscles, or it could be any kind of muscle. Okay. Fabrication of nano, micro, depending on what size you are going for the aspect ratio, nano, micro cantilever devices. Cantilever devices to study muscle force generation, muscle diagnostic. Okay. So, now what is really a cantilever? So, all of you have must been at some point or other into a swimming pool, or you must have seen a swimming pool in television or internet or somewhere. So, in a swimming pool you must have seen the diving boards. Okay. So, you have these diving boards where people kind of jump. So, if you look at a diving board, it is something like this. There is a plank popping out like this. Okay. So, this is how the plank is being attached, and this plank can move like this as well as this. So, the person stands here or maybe slightly closer here, the plank will be vibrating at a particular frequency, right? And of course, you are having a person is jumping water out here, and the plank is at a height. So, essentially this is a classic situation of a cantilever. Now, this you have seen. Now, think of it, if I could make such cantilevers in a micro dimension in order to measure forces of nanonewton, femtonewton, and those kind of stuff. Now, what one does? Now, this is is from where you can understand the whole perspective. Now I am reducing the size, okay, aspect ratio. Now what I will do, I will take a block like this. I am going on a top down approach, okay. So for example, I take a silicon wafer like this. It could be made on silicon, it could be made on PDMS, polydimethoxy sulfoxide. You guys can please look into these kind of compounds. PDMS is, I think it's Dow Corning's patented compound, polydimethoxy sulfoxide, silicon wafer. So you take a wafer like this, okay, something like this. Now what you do, you have to have a mask which will say what kind of shape you are asking. Say for example, talking about a mask, suppose I have a mask like this, array of, suppose I want an array of cantilevers like this. Okay. Now I am shading the part which are solid and the one which are not solid. Okay. Okay. So now let me shade it fully. That will kind of give you an idea how this kind of fabrication happens. Okay. Now here I have a mask. Oh shit. Okay. Okay. Now I want this pattern to be made on this block. What I will do, I will keep this mask. So say for example, I have this thin wafer of a certain thickness. Now this also has to be determined what will be the height from the base I want. Okay. So the wafer what I am using, a silicon wafer whatever I am using, or a block whatever I am using, I have to decide what should be the thickness of it. Okay. Now what I will do, I will keep the mask on top of it, something like this. I'm putting the mask in black now. And the wafer is underneath. Now using either a chemical technique or a laser based techniques, lithography technique, I am going to etch all those parts where I am putting a red. I am going to eat away this part. I am going to ensure by using certain chemicals or something, I will leave the part which is in black line and all this part will be etched out. So what I will be obtaining is something like this, exactly like this, where I will have an array of cantilevers sitting like this. Is a side view I am drawing. Okay. And of course, the depth you have decided depending on the thickness of the block. This is precisely one of the top down approach by which micro cantilevers are being made. So this kind of give you an idea. When we talked pretty early that top down approach, you are breaking the stuff, you are kind of bringing down the size and all those things. Now depending on the size of the mask, whatever is provided, you can actually decide what dimension, what aspect ratio of cantilever you are asking for. You can go to the micron range, you can even go down to nano range, you can go to even 100 nanometer range. It's all up to you what kind of things you want to diagnose. In case of muscles, generally we go for a micron range. Okay. Now, how we do the diagnostics? Now, after reaching to this point, what you do? You allow the muscle cells to grow on top of these cantilevers in order for them to form myotubes. And as I told you, the myotubes are the smallest unit of muscle. So, once the myotube grows on top of it like this, so for example, you have the myotubes going like this. So, these myotubes will start generating force. So, they will be contracting either in the presence of some drug or something, or all by themselves at times. Okay. Once these myotubes will contract, what will happen? Exactly the same situation as I told you. If this is a myotube sitting on top of it, this cantilever is going to vibrate at a certain frequency. Okay. Now, if you could make an arrangement something like this. Say for example, I am just taking one cantilever into the picture for the diagnostic purpose. So, for example, if this is a myotube which is on top of the cantilever and this is the cantilever. Now, this cantilever is vibrating at a particular frequency. Fine. Now, from here underneath, you make an arrangement to bounce a laser beam like this somewhere, maybe here or here, depending on. You are bouncing a laser in this direction on the cantilever when there are no muscle, no myotube. That laser beam will bounce out from here. Okay. Now, this angle, what the laser beam is making with the cantilever, is going to change if the cantilever beam is going down. Based on that angle, there is something called, please look into the literature on this, Stoney's equation. Stoney's equation is almost 100 years old now, or more than 100 years old. Stoney's equation will help you to calculate the force what is being generated out there. So, now if you know the force, then depending on what kind of drug molecule or what kind of pathological situation the muscle is, or what kind of material is working on muscle, you have a very simple tool by virtue of which you can quantify the force generated by the muscle. Now, you see, when you talk about diagnostics of using all kinds of nano tools, if you remember, when we talk about all the applications out here, diagnostic tools, told you about force generation studies, force generation studies of muscle using micro/nano cantilever. So, these are some of the very interesting emerging tools which are coming very handy in the animal industry if they are being used in a particular fashion. Okay. So, I will provide you the relevant literature which will kind of help you to appreciate that these are already proven thing, and much of the in vitro diagnostics are relying on these kind of tools. So, this is where I'm going to close, and we'll move further in this line with different kind of other tools where nanotechnology is coming pretty handy in the animal production. Thank you.